My dear students, in this class, let us learn about an important protozoan parasite by name Entamoeba histolytica. Entamoeba histolytica is a protozoan parasite that is present in the large intestine of the human being. Large intestine contains two parts, namely colon and rectum. Normally, this Entamoeba histolytica resides in the colon of human being, and it belongs to the class Rhizopoda. It is a monogenetic parasite that is going to complete its life cycle in a single host, namely human being. And the mode of infection is through contamination. Only source of infection is taking contaminated food and water. And it is very rampant and prevalent in rural India and in places where there is much population, where there is high population, and where there is very poor drainage system. Entamoeba histolytica causes amoebiasis or amoebic dysentery. It secretes one enzyme called histolysin, which ruptures the intestinal mucosa. Because when Entamoeba histolytica is present in the intestine, because it is rupturing the intestinal mucosa by secretion of histolysin enzyme, intestine will develop ulcers, and these ulcers start bleeding, and the blood comes out through stools. So, bloody stools or stools loaded with blood is the symptom of amoebiasis. And Entamoeba histolytica exists in three forms, namely Trophozoite, pre-cystic stage, and cystic stage. So let me repeat, Entamoeba histolytica is a protozoan parasite belonging to the class Rhizopoda. It is a monogenetic parasite completing its life cycle in a single host, namely human being. It causes amoebic dysentery or amoebiasis. It spreads through contaminated food and water. It is present in the large intestine. It secretes an enzyme histolysin that dissolves the intestinal mucosa. It exists in three distinct stages, namely trophozoite, precystic, and cystic stages. Trophozoite. In this diagram, you are seeing the trophozoite of Entamoeba histolytica. It is a motile stage, active stage, feeding stage, and pathogenic stage. The disease amoebiasis is caused by this trophozoite stage because it feeds on intestinal mucosa. And the body of trophozoite is covered by plasma membrane. It's covered by plasma membrane. And here you can see one pseudopodium, which is a finger-like projection. And this finger-like pseudopodium is called lobopodium. Finger-like pseudopodia or pseudopodium is called lobopodium. So what is lobopodia? Lobopodia are the finger-like pseudopodia that are present in certain protozoans like Entamoeba histolytica. And contractile vacuole is absent because Entamoeba histolytica is present in isotonic medium. Coming to the food vacuole, as I already told that this trophozoite stage feeds on intestinal mucosa through holozoic mode of nutrition. Trophozoite is a feeding stage that exhibits holozoic mode of nutrition, and it feeds on intestinal mucosa by secreting one enzyme called histolysin. When the intestinal mucosa and intestinal wall are ruptured, automatically the RBC along with the intestinal bacteria will enter into the food vacuole of this trophozoite. As a result, if trophozoite stage is observed, the food vacuole is loaded with RBC and intestinal bacteria. This is one of the most important characteristic features of Entamoeba histolytica. Another one is the nucleus is there. Here you can see the nucleus, which is like cycle wheel or cart wheel. Cart wheel shaped nucleus is another important characteristic feature of Entamoeba histolytica. How this is formed? Which structures give this appearance, this cartwheel like appearance to the nucleus of the Entamoeba histolytica? We will see now. So here, this is the nucleus and this is the nuclear membrane. And at the center of the nucleus, there is one structure called endosome. And below the nuclear membrane, there are certain bead like granules, and these granules are called chromatin granules. These chromatin granules are linked to the endosome by chromatin filaments, and all these structures together give a cart wheel like appearance or cycle wheel like appearance to the nucleus of the Entamoeba histolytica. So, this is another important characteristic feature. So, let me repeat: the presence of food vacuoles loaded with RBC and intestinal bacteria and the presence of cart wheel shaped nucleus are the two important identifying features and marks, diagnostic marks of Entamoeba histolytica trophozoite stage. And this trophozoite stage feeds, it goes on feeding. Of course, it performs binary fission and daughter trophozoites are formed. So, after repeated divisions, suddenly it enters into precystic stage. And all these characters I already mentioned. Single finger like lobopodium is present, contractile vacuole is absent, body is covered by plasmalemma, food vacuole is loaded with RBC and intestinal bacteria, cart wheel shaped nucleus is present. And one more point is mitochondria are absent in Entamoeba histolytica. The reason is this Entamoeba histolytica, it is present in the intestine where the probability of getting oxygen is very, very remote. Because it is residing in the intestine where the probability of getting oxygen is very remote and very rare, it has to perform anaerobic mode of respiration. As a result, there are no mitochondria in Entamoeba histolytica. The absence of mitochondria clearly confirms the obligate anaerobic nature of this particular parasite. Mode of nutrition is animal mode of nutrition, namely holozoic. And now, after voracious feeding and after continuous binary fission, suddenly the trophozoite changes its behavior and it enters into precystic stage. And precystic stage is non-feeding, non-pathogenic stage, non-motile form. And glycogen granules and chromatid bars are found in precystic stage. Glycogen granules are nothing but reserve food material that are present in the precystic stage. Chromatid bars are made up of ribonucleoprotein. And after some time, these precystic stages are converted into cystic stage. Cystic stage is round in shape and they form cysts, and this process of formation of cyst is called encystation, and this cyst is highly resistant to the environmental factors. And once the cyst is formed, inside the cystic stage, the nucleus undergoes two successive mitotic division forming four daughter nuclei. And the cyst that contains four nuclei is called tetranucleate cyst. And this tetranucleate cyst is the infective stage to the next human being. And these tetranucleate cysts come out through fecal matter of the human being. This is why open defecation is not advisable. It's not advisable. And eating contaminated food outside is also not advisable. So, tetranucleate cysts come out through fecal matter, and this fecal matter enters into the soil, of course, and houseflies act as transmitting agents. Whenever the housefly come and alight on the fecal matter, normally they will be going, houseflies go on a picnic and they come and visit nearby ponds, and defecation occurs in pond area and they alight on the fecal matter which contain tetranucleate cells, and these tetranucleate cells get attached to the body of houseflies, and these houseflies will come to your house, will go to the hotel in which you are dining, of course, and they eat on your pizza, burger, everything, and they simply drop the tetranucleate cells on these edible items. That means all these edibles and eatables should be protected from the houseflies. Otherwise, they may get contaminated with tetranucleate cysts. And now, along with the food and along with the water, you take this tetranucleate cysts into your body. And now, this tetranucleate cysts enter into your intestine. And once it enters into the intestine of another human being, the cyst is dissolved by the action of trypsin enzyme. And the very fact that the trypsin enzyme is digesting the cyst, it indicates that cyst is made up of protein. Now, excitation takes place. After that, metacystic forms are released. The nucleus, the four nuclei are there in the cystic stage, which are released, of course, in the form of metacystic forms. And these four nuclei undergo one division each, forming 8 nuclei. And around this 8 nuclei, some protoplasm gets accumulated and they get separated, forming daughter entamoeba. Like that, infection continues, and this daughter entamoeba will get converted again into trophozoite, and the rest of the life cycle repeats. That's all. So let me show you in the schematic diagram. Trophozoite is there, trophozoite will be there. Later it undergoes binary fission producing daughter entamoeba. Some young ones get transformed into precystic stage, later into cystic stage. Tetranucleate cysts are formed, of course, and they come out along with the fecal matter. New host will be accessible because of the house flies, and by taking contaminated food and water, new human being will get infected. Once the tetranucleate cells reach the human intestine, they get ruptured, the cyst gets ruptured because of the trypsin activity, releasing metastasis, and ultimately 8 daughter metastasis will be released. And this is a diagrammatic representation, of course. And the disease is called amoebiasis or amoebic dysentery. Sometimes the intestine will get swollen. This is called peritonitis. Sometimes they may get settled in liver, and some people may not exhibit all these symptoms. They are asymptomatic carriers, of course. Whatever may be, the treatment is there. There is a very effective treatment, and the drug that is used for the treatment of this amoebiasis is metronidazole. So metronidazole is the most effective drug that is used to treat amoebiasis. So that is about Entamoeba histolytica. In the next class, we will meet with some other topic. Thank you very much.